हमें सिलेबस के ऊपर। हाँ संजय सर।
हाँ बोलिए।
मैं कहा एक डिस्कशन हमें ना सिलेबस के ऊपर करना चाहिए कि जो चेंजेस किए हैं वो
हम्म।
कितने प्रैक्टिकल है और वो टीचिंग लर्निंग में कुछ चेंजेस हो भी रहे हैं या नहीं हो रहे।
ठीक है, उसके लिए भी हम लोग कहीं बैठ जाए, है ना, कभी ऑनलाइन। ठीक है।
हाँ बिल्कुल।
हाँ, रहेंगे, प्रशांत सर भी रहेंगे, डॉक्टर रमेश जी भी रहेंगे, है ना, डॉक्टर नमता जी को ज्वाइन किया जाएगा।
हाँ हाँ बिल्कुल बिल्कुल।
एक बार करते हैं डिस्कस। करना चाहिए, आपस में करना चाहिए। और आपके यहाँ क्या चल रहा है, हमारे यहाँ क्या चल रहा है।
क्योंकि नहीं मेरे को तो ऐसा लगता है कि सीक्वेंस बिगड़ गया है पढ़ाने वाला। स्प्लिट हो गई हैं चीजें तीन टर्म्स में ना कि फर्स्ट टर्म में आपको एक पार्ट का कुछ पोर्शन पढ़ाना है 30%, फिर 30% पढ़ाना है। कॉन्टिन्यूटी नहीं बन पाती। नम्रता मैडम नहीं जुड़ेंगे क्या?
कौन?
नम्रता मैडम नहीं आएंगे क्या?
और डॉक्टर रमेश जी को मालूम होगा। रमेश जी, डॉक्टर रमेश जी, डॉक्टर रमेश जी। डॉक्टर रमेश जी कहाँ चले गए? तो डॉक्टर पीयूष जी स्टार्ट किया जाए।
हाँ सर स्टार्ट करते हैं।
कि 8-5 हो गए हैं, है ना? क्या? और
Namaste and a very warm welcome to all. Myself, Dr. Shivakshi Sharma, Senior Resident in the Department of Rasashastra A, Bhaishajya, Kalpana, IMS BHU, and I feel deeply honored to serve as the coordinator of today's national webinar by Vishwa Ayurveda Parishad. Um, on behalf of Vishwa Ayurveda Parishad and our organizing committee, I extend my heartfelt greeting to all the dignitaries, invited speaker, respected scholars, Vaidyas, faculty members, students, and Ayurveda enthusiasts who have joined us from across the country today. Now, moving forward to invoke the divine blessings at the beginning of this auspicious gathering, I request Miss Namrata Singh and Miss Sneha Lata Singh, UG students from Pilibhit, to present the Dhanvantari Vandana. Over to you, girls.
नमस्ते सर, नमस्ते नमस्ते मैम। नमामि धन्वन्तरिमादिदेवं सुरासुरैर्वन्दितपादपद्यं लोके जरारुग्भयमृत्युनाशं धातारमीशं विविधौषधीनां च व्योमवातावनिवारिवह्निं पंच प्रपंचात्मकं देहभाजं संताप संपात जरा ज्वरांतकं नमामि धन्वन्तरिमादिदेवं नमामि धन्वन्तरिमादिदेवं नवीन नीलं मुदकान्तिकान्तं शान्तं हरेव द्वादशमाख्यमूर्तिं पूर्तिं शतानां सुमनोरथानां नमामि धन्वन्तरिमादिदेवम् नमामि धन्वन्तरिमादिदेवम्.
Thank you for that soulful and serene rendition. May this Vanna brings positivity and sacred energy to our today's deliberation. To formally begin this session, I now take the pleasure of inviting Dr. Sanjay Agrawal, sir, from NIA Jaipur, to deliver the welcome address and set the tone for today's event. Over to you, sir.
Thank you, Dr. Shivakshi Sharma ji. On behalf of Vishwa Ayurveda Parishad, Rasashastra, I welcome to all of the participants, our senior, some renowned personality from the across the India, and Dr. Bhaishajya Bhattacharya ji. She joined also from the USA. Once again, I welcome to all of you in this webinar. Today the topic is very interesting. It is going to be delivered by the Dr. Prashant Bedarkar, sir. Approach to trend of safety evaluation of Rasaushadhi A, call for critical review. As we know, the Rasaushadhi, there is always a burning issue regarding the safety of Rasaushadhi, whether it is safe or unsafe. There are lots of study which was conducted in the different institute regarding the safety study on the basis of acute toxicity study, subacute toxicity, and the chronic toxicity study. Generally, what I feel, the report from the various institutes, from the various journal, various article, Rasaushadhi are safe on the basis of acute and subacute study. But regarding the characterization of Rasaushadhi, it may differ from the institute to institute or from the different colleges, whatsoever reported in the various journals. We know that our Rasaushadhi, the herbal mineral compound, which is very effective, when it is quoted in the Sensar Sangra, Sadhesu Vesjam, Sarvamiritam Tatavedina, Asadesa Pidatabhyo, Rasavata Srishtam Uchyate. For the curable disease, there are so many medicines, but Rasaushadhi act also upon which are not curable through the Rasaushadhi. So this is a great importance of our herbal mineral compound. But the worldwide, it is not accepted due to the safety profile, safety study. So in this today webinar, we bring with a very interesting topic which will be delivered by the Dr. Prashant Bedarkar, sir. He is a professor and most dedicated person in the Department of Rasashastra, Jamnagar. So I hope sir will focus on the safety issue and what are the recent study report which has been conducted on the various metal mineral compounds. Not taking much more time, now I once again I welcome Dr. Prashant Bedarkar, sir. Now over to Dr. Shivakshi Sharma.
Thank you, Dr. Sanjay, sir. Now, moving forward, I would like to introduce our esteemed guest speaker for today's session. I would like to invite Dr. Piyush Chaudhary, sir, from Sonipat. Over to you, sir.
Thank you, Rakshi. To introduce our today's speaker, Dr. Prashant Bedarkar, sir. He is an eminent scholar, researcher, and teacher in the field of Rasashastra. and is currently serving as professor at ITRA Jamnagar.
डॉक्टर पीयूष जी माफ कीजिए, हमारे ही लोग हैं, 20-25 ही लोग हैं, बस हम बहुत छोटे से हैं, तो IPGT से that is just sufficient. Sorry for interruption. Let me just start the subject. We have very few time, and I will not engage you not more than, we have reached already 8:15. So I will not take more than 40 minutes, I think so. Or I will be very top to the bottom and very very unbiased, and I will try to put forth the facts. धन्यवाद आपका छोटा सा इंट्रोडक्शन देने के लिए. So that is more than sufficient for me to engage with ITRA. That's all. Okay, thank you. By breaking protocol, I first of all pay my regards and thanks to Vishwa Ayurveda Parishad and all eminent authorities of Parishad, Namrata Doshi Madam, Sanjay Agrawal sir, P.S. Chaudhary ji, Ramesh Gupta sir, and many more. So with this, I will not, it is not a teaching and something else and just presenting, and I will not be talking very fascinating terms, and I will not present in some manner which looks very fine and good. I will not try to do this. So I will pave a basic pathway. I will put forth, these are my personal opinions. The safety issue is a topic of debate as described by Chaudhary Sanjay Agrawal sir. It is a policy matter. It involves commercialization. It is a and it should not be taken as lightly as we are taking and we are treating forwards. When we are in a house, it is very fascinating, and it is very very much comfort. We are in very much comfortable zone to say that Rasaushadhis are safe, Ayurvedic medicines are safe, and everything is safe. Here at ITRA, we are prescribing mercurials, 35 mercurials, and turnover of 135 kg annually. Likewise, huge turnover, more than 1,500 patients, and 33% is rate of prescription of Rasaushadhis containing mercurials, which was studied in 2017 over 2,000 patients over 5 days period. So it doesn't mean we have a lot of documentation of safety, and when we are talking in between in-house, it sounds good that Rasaushadhis are safe, everything is safe, but we should retrograde. We should put forth facts, and we should analyze what status, what documentation we have generated, what we are doing, and unbiased with unbiased manner. We should look to the process, what we have done and what we are going to do. So the few basic questions we do have very frequently: aren't Rasaushadhis safe? When we talk in-house, yes, very, very certainly those are safe. There is no problem. We have sufficient documentation. We have sufficient practices. As I told, we prescribed 25 years back Garbhapal Ras, most vulnerable condition, developmental age. We have clinical documentations. We have third generation reproductive study more than 30 years ago by BHU. So, but still, it doesn't, it is not sufficient. We do have, I agree that we do have a lot of data, but still we need, our presentations are not in a proper manner, in a proper site, in a proper journal. There are a lot of lacunas, and still there is more need to work upon. So is it needed to evaluate safety of Rasaushadhi through either toxicity or other methods? Then again the question is, aren't practices studied, conducted, documentations available regarding safety of Rasaushadhi sufficient to state their safety? Then there is, everybody knows, there is paucity in standardization, product nature, ADME, and all those things. And we take this consideration. I may discuss it in a few last slides, but we usually take it into consideration. We know there is a lot of problem with standardization. We do have monographs, we do have AFI, we do have strategic protocols, but in spite of that, this is a very, very much hurdle. and very painful thing that we are still lacking, very severely lacking in this sector. And whatever the studies those are conducted, if we don't have a data of reproducibility, assurance, precision, accuracy, and if there is missing data, the study conducted does have very little significance. I know that if you are going through one protocol, if you already have developed very stringent protocol for standardization, pharmaceutical standardization, analytical standardization, it is not possible that everybody will adopt it. There are a lot of parallel ways of its preparation. Lot of pharmacies are preparing. Lot of you can follow AFI, you can follow all 54 schedule books, and there could be lot of parallel protocols. But still, development of monograph, following of one standard protocol is a need of time. And to my knowledge, it may be, I am very small fry, and I should not decline the contribution of stalwarts who have contributed almost before my birth. They have done very significant works before my birth. Also, they have conducted even Paina Sanskar and metallurgy. BHU people have worked a lot of things. Namrata Joshi madam also have contributed a lot of. Anand Chaudhary sir for Loha. Lot of protocols are there, and good documentation, quality documentation is there. I am nowhere to stand before, and there may be many faces who have heard my name first time. I agree. So I'm no one in this sector, but still, as per my knowledge, I didn't found a single pharmaceutical protocol which is published, which is devoid of good quality missing data. So there is still need. There is no reproducibility. There is no accuracy. We lack these things, and we must accept. And until and unless, if you are working with, say, if I am working with pharmacology for clinical study, it is okay. But if you are working on safety, we cannot compromise with product standardization, because every other pharmacological evaluation, clinical evaluation, and everything will be, this will be the base. They will try to adhere to this protocol, what was used in this safety study. So until and unless there is precise documentation of pharmaceutical and analytical standardization, I have taken more than, I think, 10 minutes rather than focusing on safety study. So this suggests that until and unless this is controlled, further claim of safety and standardization, you cannot claim what people are conducting. You have same margin of safety. If we claim that here we have prepared more than 35 batches of Samir Pannagras, and the scholar have, yesterday only there was discussion, only 2 XRDs were there, and they were not matching. We were having all product characteristics same, and all yield, repeatability in yield, and all those things, but 2 XRDs, those were not matching. So it gives a serious significance that we must retrospect ourselves. So we know our product nature, ADME, there are a lot of problems with this, which are there is paucity, and there are, which raises concerns for evaluation of safety and need of safety evaluation. So duration of administration is also a question, and on this is the only basis where we are lacking and we are not proceeding further. There are many, might be many reasons for why we are not proceeding more than 3 months toxicity study, repeated toxicity study more than 90 days. 1 or 2 there are 6-month studies, but we are not exceeding 90-day study. And this protocol we have adopted very few couple of years ago, not more than a decade. So and we have only handful studies with this duration, but people are prescribing it over years together, for 5 years, 10 years, and more than this. People are documentations of for cancer treatment, and as they get challenging results, they continue on. There are people who are consuming as a OTC, like for SWAS, for Amlapitta Sutshekar, and SKR, Swasputar. Likewise, people are taking and there is no control over, although there are guidelines, recommendations by on ICE website also, but still we don't have a proper control over OTC medicines and prescribed medicines. We don't have, we do have recommendations in situ, but not still not regulations by gazette notifications and all. But and it gives it paves pathway for evaluation of long-term safety evaluation, and why we are restricting ourselves to only 9 months, why not 1 year? And in fact the available protocols are not suitable for us. We should have higher standard than what OECD states, and we are not achieving that only. There is no study which is fully in compliance with OECD. And I don't know, there are a lot of debates, as I suggested. It is not necessary. We are not bounded to follow ICH, OECD. We do have guidelines by CCRS. There was draft on ISH 170, which is outdated, then replaced with miscellaneous advertisement. And then there is a lot of debates. It is never came into force. But we do have several guidelines. We do have D&C also. There is a lot of debate that what should be followed. But to my knowledge, we should follow higher standards than this, and logics for that we will be discussing in few next slides. And as there is global propagation, we must have a quality documentation of safety. So acknowledgement of, we know the Rasaushadhis are safe, or Ayurvedic medicines are safe, but acknowledgement of their heavy metal, acknowledgement of heavy metal toxicity has wider, it is acknowledgement of and conveying safety of Rasaushadhi to common community, to scientific community, to globally, is very, very, very difficult task, because the counterpart, heavy metal toxicity, its acknowledgement, it has wider acceptance, wider publicity, strong scientific support, huge database of quality studies, because it is matter of global trade, economy, health, and human rights, involvement of many regulatory bodies, association of commercial vendors and entrepreneurs, and studies conducted by conventional scientists, basic expert scientists, incorporating model evaluation tools, and with recently approved protocols. And they are working since ages, since centuries. They are working with this heavy metal toxicity, and they have lot of documentation. With arsenic, more than 12 models, and I'm taking one species as one model. So more than 12 models for organoteins, more than 14 models they have worked with. So against this, if we are claiming that my lead in this form is safer, the form we don't know how we are administering, we have not decoded it scientifically, we have not put forth, we have not reproduced repeatability, and then how we can, when there is a data of decades in different models, in in vitro studies, lot of in vivo models, and clinical documentation. And if we are not going, we will discuss it, that alternative protocols we will have to opt. We will have to focus on, there is no way that we can keep it aside. Clinical documentation is needed, clinical documentation of safety is needed. And if we are not doing, against this, people are there, they have opened their websites, people in toxicology, those are working, people with NIH department, yes, those are working. They have made open platform. Anybody consuming lead from traditional medication, they can report it there, and they have made these platforms available. So rather than developing data and controversies against traditional medications and Rasaushadhis, it is we should parallelly develop the data of their consumption, distribution studies, prescriptions studies, and evaluation in vulnerable groups, and likewise we should build up parallel data, which should be thousands of times and lakh of times more than that. And we do have potential here. Every day more than 330 patients are receiving at all over country, more than thousands of patients receiving. Why it is not documented? If you protocols are there, it is very much possible. But it is need of time that we should pile up it, because animal studies and evaluation, they have several lacunae in terms, because there is basic difference in between contemporary medication and Ayurveda medicine. So we will specifically Rasaushadhi, which we will discuss in next slide. Everybody knows that level of evidence, clinical documentation, even a single expert opinion has more level of documentation and value rather than a well-designed animal in vitro study. So we do have this data, and we are underutilizing this potential. So the basic difference in contemporary medicine and Ayurveda medicine is that they have developed, they have evaluated this safety in a few models, and in few 4 or 5 rats. In a few models, we have the data of centuries. We do have practices ongoing, retrospective studies. We can have many subjects, many administrations, long-term clinical administrations, and in different setups we could have data along with different drug administration, with different protocols, with different adjuvants, with different dosage forms, in different methods of preparation, in different variants of same formulation, in different manner if it is prescribed. In different vulnerable groups, and in different same group with same patient with different complications. Likewise, we do have a lot of data. We do have pharmacovigilance centers. We do have a lot of educational institutes from where the data can be generated, and wide range of drug food combinations we are practicing. This data is not available with contemporary medications, and albeit we accept that there is no doubt that Rasaushadhis are safer. We do have after this protocol development of a new molecule, after 10,000 compound screening, only one is approved, and you can note the its withdrawal. If you subscribe the WHO drug alert, every day you will find one drug withdrawal and safety concern, even for food products, even for milk products, dairy products, fish, marine products. Daily you will get one alert that this has been banned, this has been withdrawn. Likewise, so there is significant withdrawal. More than 644 drugs were withdrawn from 50 to 14, and the market sustenance is very, very poor. So in respect to that, your drugs are very versatile. Nothing has been withdrawn. Still we are practicing, and high versatility of Ayurveda medicines. You triturate in many manner. You do heating operations in many protocols, right from 30 degrees to 650,000 degrees. You work up with you have multiple. You do sometimes boiling, mixing with different pH, with different combinations. You do practice polypharmacy, and sometimes you do pak in molten gandhaks. In gaseous form you treat it, and after that you use in multiple combinations, in multiple ways. It gives versatility that in spite of that, if there are no significant fatalities, significant concerns with neurotoxicity, hepatotoxicity, nephrotoxicity, it is there is no doubt that your drugs are safer. But if you see, 10 times more than administration of therapeutic administration, 10 times and 5 times administration, we don't get anything in tissue histopathology and in basic vital parameters and biochemical markers of LFT, KFT, and hematology. Likewise, in every study, those are done all across studies, and we don't get anything even in 5 times, 10 times. Forget about therapeutic dose. And therapeutic dose of, say, if you are working with one bhasma, so it is a component of some Rasaushadhi, so it is there 1/10 and sometimes 1/50th times there in the Rasaushadhi, and 10 times of this therapeutic dose, that doesn't gives any toxicity. So we are having very high therapeutic index. You cannot find more than 2 times or 5 times. It is considered as toxicity, and that is taken as toxicity in protocols. Why? Because more than that, there is no safety. and you demonstrate it. So there is no question. So versatility, sometimes we don't prescribe, we don't follow protocols of Kshetrikaran, Pathya, Anupan. Very less occasionally we take it into consideration. We never ask a lady in a fertile age whether you are going to conceive, and we prescribe medication for conceivation. Several Rasaushadhis we prescribe for conceiving, and after that she conceives. That means highest. We have talked about Garbhapal Ras, but when you are prescribing 1 year Rasaushadhis before conceiving, and then there are no fetal adverse outcomes, if those were, these practices will have been outdated. So we never ask a lady whether you are going to conceive. And equally there in this 33% prescriptions, there are equally ladies are also there in fertile age. So you never ask them. So it is proof of safety. We don't follow all classical guidelines, and what is optimally recommended, we hardly follow. We never use Ashtasanskarit, Parat, Zarit, and all those things. But in spite of that, we don't get. We hardly consider alcohol drug interaction and all those things, and Kakarashtak and all. People are consuming Nagarveli Patra, Karkati, Karvelak. Those who are on those who are diabetic patients, those are consuming Amla Varga Dravya frequently along with Tamra Basma. Whole South is practicing daily. I don't forget what they consume, sambar and all those things. Here in Gujarat, everybody consumes takra. Without takra, there is no. So there are a lot of, in spite of that, there is high versatility towards safety, which are contraindicated in spite of that. And polypharmacy, there are several formulations, 50 combinations, 60 combinations all together, but we don't get drug-drug interactions. So CCRS had done a lot of work, more than 12 books, more than 15 Rasaushadhis. Here in IPGT and RA, we have conducted more than 30 Bhasmas, and more than genotoxicity study, more than 10 genotoxicity studies. And if you see the data, 5 times TED, 10 times TED, we don't get any safety concerns. Forget about TED. So lot of documentations we do have. We do have monographs. These slides are very age old. There are many further updations in this. There are documentations, 11 acute toxicity studies by only Dhatu Varga Dravya. It was screened for only Dhatu Varga Bhasma. 11 toxicity toxicity minerals, Rasa Varga Dravya. The rest of the things are excluded in this study. So 11 drugs toxicity, acute toxicity study. Overall screening, more than 75 repeated dose toxicity studies are available online. And along with Shar, Rasaushadhi, and Vishaushadhi, more than 13 toxicity studies. And along with Shar, Vishaushadhi, there is high vulnerability for toxicity. If you are working, if you are using Rasaushadhi, along with this, there are ingredients Rasaushadhi containing Shar and Vishokvish. There is more propensity for toxicity, and more than 35 such studies are available in the public domain. So we do have monographs also, Raskarpur monograph, detailed studies, multiple studies, Nagbasma, and at RSBK department ITRA, we do have also monograph of safety studies and clinical safety studies of Rasaushadhis, and their contemporary pharmaceutical units are also doing contributing to this. So there are many reasons why we do have insufficient. In spite of that, I know a lot of work have been done and a lot of quality work have been done, but still there is, I agree that there is insufficient documentation. There is no still optimal documentation is needed. Optimum data, quality data is still needed. And why there is insufficient documentation, and we lack, we are not having quality documentation which is expected and needed. There are a lot of concerns, because it is not mandatory for us it is optional to do safety studies. Even there is no need by legally in country you don't have you don't need it. But 11 countries around India, if you want to, where there is you can export as Ayurveda medicine, Sri Lanka, Thailand, likewise Japan, this Nepal, likewise nearby. If you are exporting, they may ask for, and legally you may be yeah, you may need it. But and that is why we are lacking. We are taking do not taking it more seriously. So recommended duration of studies we are restricted to 6 to 9 months only. And we can go far for chronic toxicity study. They are working on 1 year basis, and it is not needed for them, because there are very few drugs which are bioaccumulation in conventional science. Their frequency of administration is once a day, or sometimes once a week. So their half-life is not more than 3 days. The highest half-life is not more than 3 days, and almost all drugs, they have half-life of more than an average of 12 hours. So they don't need it, but still they recommend for 1 year toxicity study. And there are a lot of needful things we will discuss in further slides. You do have bioaccumulation, and which is well acknowledged, and it can be there for 25 years, for decades it can be there. And then it is need for us. People ask for this long-term safety, recovery, and long-term studies. They don't rely on 3-month administration and 6 months administration. Okay, you have administered only 3 months, but what is the follow-up of recovery? They ask for recovery for 1 year, for 6 months, not merely for 3 months. So this data is needed, and otherwise scientific community will not accept this. So you do have you could have bioaccumulation in tissues, and delayed effects are there. So there is probability for delayed toxicity, and thereby we need more stringent evaluation and customization of toxicity. But we do have lacuna. You cannot administer in a rat more than 1 year. Lifespan is 2 months, and for less than smaller animals than that, then the life cycle is more than this, less than this. And if you want to work upon higher animals, then there are ethical concerns, and there are limitations, cost, and there are a lot of concerns to work up with higher animals. And that is why, and how many formulations, in how many ways, in how many adjuvants, in how many clinical setups you can work up. You can work up with only one protocol, and it gives pathway, it paves pathway and need for evaluation of therapeutic drug monitoring and clinical safety, need for documentation of clinical safety documentation. But still, what we are adopting, it needs updation in it also. So because this the heavy metal toxicity, which is classical, you may not expect only the safety. If you just want to evaluate the heavy metal toxicity of Nag Bhasma in terms of heavy metal toxicity of lead, it might not be sufficient, because your toxicity protocol, your toxicity manifestation might be different from that. If you are giving simultaneously lead and other Vishavarga Dravya, which have equal and opposite actions on, say, CNS, PNS, like if you are using say Dhatur, if you are using say Kuchila, some are CNS suppressant, some are activating. So you cannot just watch for, you cannot expect this presentation. You have to have holistic evaluation. You cannot rely on one presentation. You have to document your own toxicity. If it exceeds in vulnerable group, if it is higher doses, a long-term toxicity, we must have our own documentation of expression of toxicity. So there are basic differences in conventional safety evaluation of conventional drugs and Ayurveda drugs. So I will not take into consideration most of the things we have taken into consideration. Only they don't expect either drug-drug interaction or drug-food interactions, or if it is well acknowledged, if it is there, but we don't know. We have to work up with this. And in safety study, evaluation of safety study, you are working with only one drug, one adjuvant, and in clinical setup, it is not the scenario. When you are working with, you are administering along with some Vishavarga Dravya, some char, a pinch of char, pinch of leaven can form different salts, and the solubility might be different, kinetic might be different. And for that only we use this, for Dipan property, Pachan property, we use this. And we expect its activity of, say, in Swaskutha Ras, we expect this 9 mg of arsenic, 9 mg of Kajali to be active, and there are a lot of documentations for that. But this 9 mg doesn't work when it is individually given. With this 9 mg of pipe shunti, pipuli powder doesn't work. When it is there in the combination, it works. So you cannot neglect this drug-drug combination, synergism, and polypharmacy, which you are practicing. You are combining drugs, mixing drugs, and giving. It could have different presentations, and there could be a lot of concerns. So it is needed to document, and you cannot just rely on safety toxicity of this basic ingredient of your component of your Rasaushadhi, that is Bhasma, Nagabhasma, or Manashila, toxicity of Manashila. No. Beyond that, it is needed, because on contemporary drug is planned and developed in a different manner, and your drug is different. The contemporary drugs, it is made to be 100% water soluble. It should be 100% bioavailable, but your drug is not a drug. It is a product. First of all, we should understand that it is a product. It could be a bioenhancer. So whatever you are administering, you are not sure it is in the form. If you are administering alpha HGS, you are not sure in serum it is going alpha HGS. In deposition tissue, it is alpha HGS. If you are administering low Bhasma in a ferric form, in a transport pool, it is in ferric form. In deposit form, it is in ferrous form. Ferrous form is bioavailable, good bioavailability, but again transformed into ferric form when it is combined to ferritin. So, but you are administering in a ferric form. Right from beginning, ferric form, poor bioavailability, but transformation needs less transformation. So there is a lot of difference. What you are administering, you don't know whether it is there in plasma in the same form, in the tissue in the same form. When you are administering concomitantly administering many things, you don't know the secondary derivatives and tertiary and derivatives and what is working. It is product. Most of the things are not soluble and not bioavailable, not 100% bioavailable, and throughout gut they may be treating. There are drug-drug interaction, there are drug-food interaction, there are expected drug-food interaction. Whereas when in your stomach, within 10 minutes, your drug is 100% soluble in contemporary medication. It is achieving peak plasma level within 30 minutes, and there is no drug-food interaction, drug-drug interaction, and they don't expect it. But you expect it. So the data of safety studies might not be sufficient when you are its clinical applicability is extrapolated. So, and because your all drugs, they play significant role. Those are enzyme regulators, those are catalysts, those are essential trace elements, those are having bioaccumulation. So there can be pre-metabolic interactions, post-metabolic interactions. So you cannot neglect, and this is our principle. If you give the same Bhasma along with different Anupan, you expect that it for different disease conditions, for different indications, you change Anupan, Sahapan, mode of administration. If you are administering in a lehal form along with a little quantity of Madhu and Ghee, there could be a sublingual administration and some nanoparticles. There may be in a systemic circulation. If you are orally administering along with quantity of Anupan is more, it will be there in a stomach. It will throughout intestine. It will go. Whatever you are eating, it will be the drug-drug interactions, drug-food interactions, and whatever the secondary derivatives will work. So you expect pre-metabolic interactions, because you expect if you are changing Anupan, Sahapan, you expect different activity. If you expect if you mix it along with different drugs, you expect different actions. So you expect this pre-metabolic interactions or post-metabolic interactions, sometimes competitive binding, transport of different proteins, which the proteins which the different metals, trace elements require different transport proteins. So they engage these proteins, they compete with your drug, your food, for binding, for metabolism. They have target allosterism. They can precipitate your drug. So there is competitive binding, there is enzyme inhibition. The enzymes needed for their metabolism, they are engaging in this enzyme. They can induce enzyme, they can be enzyme inducer, they can inhibit the enzymes. Lot of alterations possibilities are there, and with these possibilities, for these possibilities you give that when you add a pinch of this Rasaushadhi, it works. Your drug, your Kastaushadhi works in a different manner. It becomes potent. It is your phenomenon. Then how you can neglect this effect? If when you are working when in a clinical setup, there can be a toxicity if you are working with higher doses in different combinations. If you are working, how you can extrapolate the data of one toxicity study to clinical setup? There is need to, and you cannot have a parallel lot of toxicity studies by changing such protocols. So it is only the answer is clinical documentation of safety through safety evaluation of protocols. So with excretion also, with changing pH of urine, they can alter renal flow and alter the excretion of contemporary concomitant administered drug. So why drug-drug interactions, and there could be a lot of drug-drug interactions along with other drugs, along with other Ayurveda drugs, other with contemporary traditional medications or contemporary medications, drug-drug interactions, and that must be documented. And when something happens, some adverse thing happens, who will be the culprit? When some adverse effect is noted, who will be the culprit? Because contemporary people, they will raise their hands. I know the plasma level. I know whether there are these adverse effects are there due to this drug, whether it causes hepatotoxicity or not. I know this, but you don't have the documentation when it is given, because you don't have data of kinetic. You don't have data of kinetic when it is given along with some other drugs, when different foods, when it is given. So the culprit, where there is no documentation, where is the lack of data, where there is paucity, where you don't know those related kinetics, those related effect, those related toxicity. If you don't know, then the first culprit is, they will say this is due to your drug. The renal failure, it is due to your drug. Hepatotoxicity, it is due to your drug. So that is why we must have data of drug-drug interaction, and otherwise there will be a limited utility of Rasaushadhi. So there is lot of scope to work up with for conventional anti-diabetic drugs, hypertensive with hypertension, whether it increases, whether it lowers the lowers blood pressure, alters your heart rate. It must be documented. So Aspartate is given along with beta blockers, calcium channel blockers, whether it further reduces heart rate, further reduces blood pressure. This documentation should be there. Otherwise, it will be, nobody will be advocating for its concomitant administration. So, as known, drug-drug interaction is widely practiced but poorly explored and poorly understood phenomena. These are from website of WHO. This statement is not from a smaller platform. So there are several legal aspects of safety evaluation. We can we have OECD guidelines, ICS, but as I mentioned, it is it may be debatable. CCRS guidelines might be sufficient for us. We may not be abide to go for others, but it is our moral duty and it is need, and contemporary people will ask for stringent parameters. If you are going globally, if you are going to scientific contemporary community, you must have data as they expect, and that is why we must improve. So recommended protocol for evaluation, you must have what contemporary people expect. They expect that of pharmacokinetic, toxicokinetic, which would, I know for toxicokinetic we have a lot of limitations, and it cannot be in the said protocol. You cannot fit, because it is not, you cannot give IV, you cannot compare their equations, you cannot fit to their equations. So for deriving this data, you need to check this along with IV administration and all. So there are limitations. What you will measure? Is it sufficient to measure only Hg? It might not be. So what you will measure? We don't know what to measure. So there are a lot of lacunas for that, but it is not that we should not, and we cannot. Lot of things can be there. The tissue distribution and there's heavy metal concerns that can be there, and it is not that there are no such data. There are data, but those are not up to the mark. We need to improve it a lot. So even post-marketing data and data in special vulnerable population, post-marketing surveillance, and even people are concerned about environmental concerns. It is safe to human. Does it mean that your excreta, your urine, the form in which it is excreted, being excreted in urine, is it safe for marine, for veterinary, for AOs, for birds, for all, for flora and fauna? So people are concerning with this, whether your saliva is safer to all those things you are using, you are excreting daily. So is it, people ask for this data. Is it safer for human? Does it mean safer for all species? No. So people ask for such fine data. The male fertility study, female fertility, separate female fertility study, developmental study. Although if you are giving
Prashant sir, please unmute yourself. You are muted. You are muted. Just unmute yourself. सर आप म्यूटेड है, म्यूट है, म्यूट करिए.
From Madhuri.
अच्छा
अभी आ गया. हाँ, ओके सर. तो पीपीटी पर जाइए. हाँ सर, स्लाइड्स दिख रही है सर?
Yes sir.
Visible and audible.
Moving, slides are moving?
Yeah.
Yes sir.
Yeah. So people ask for developmental toxicity. ये चल रही थी फाइल, लास्ट में म्यूट हुए तो ये चल रही थी स्लाइड सर?
बिल्कुल चल रही है.
हाँ, so developmental toxicity study, people ask for, if you are giving for cerebral palsy and in pediatric, if you are giving Rasaushadhi, the developmental toxicity is also needed. Carcinogenicity, we consider that carcinogenicity and immunotoxicity study and endocrinal toxicity study. There are a lot of debates. Endocrinal toxicity study, it is well known fact that lead, mercury, it works with, it interferes with all endocrines. It is well known fact. So there are a lot of things we don't know, and why we are not doing this. There are a lot of things, because if you do this toxicity study, if you are working with endocrines, something will disbalance, because it is drug. It interferes with this. It is very critical issue to put forth and document in a very proper manner, because in a volunteer in a healthy state, we have to document that in a healthy state it is not interfering, and in a pathological state it is working. The same thing as if you are giving amlaki, say half liter of juice of amlaki, it is safer. But if you are working pinch of only amlaki powder, then also it is reducing blood sugars, but half liter of amlaki soda doesn't causes hypoglycemia. The same thing is with guduchi, same thing with haridra and all those things. So we have to put forth in a proper manner. Otherwise, why we are not doing this? Otherwise, there will be alterations, several alterations in endocrines and immune system. It interferes. It's well-known fact. Everything interferes with this, but it is to be put forth in a proper manner. So, but people ask for this, that for immunotoxicity, for there are concerns. You cannot say that it doesn't work with. We claim for rasayan, we claim for adaptogenicity, we claim for epigenetic changes, we claim that it works with basic pathology of diabetes. It doesn't only reduces blood sugar, it corrects the pathology. Because Trivanga Bhasma we are giving, you cannot play on both the sides. Trivanga Bhasma, after stopping medication after 1 month, after Avrak administration after stopping 1 month, after Low Bhasma after stopping 1 month of medications, we are having good control, glycemic control, and even highest glycemic control after stopping, without giving any medication. There is no single drug in market which can have this sustained glycemic control. So people take this as a proof of toxicity. It is well known fact that lead interferes with carbohydrate metabolism. It is not a new thing. People know that. And people take it as a tool for toxicity. They claim it is toxicity. So we have to, that is why it is needed. We have to put forth the data of delayed toxicity. We have to put forth, it doesn't reduces only blood sugar, it works on basic pathology, insulin sensitivity, glucose uptake, until and unless we put forth for this data and long-term safety evaluation after administration medication, long-term follow-up, until and unless we put forth the data of long-term follow-up, it is unacceptable. And this long-term follow-up for 1 year, 2 year, decades, it is not possible with animal studies, because there is aging. Mode of dying can be there, but there are no such protocols to record mode of dying and all. So because there are a lot of concerns, you have to develop methods, and method development is not an easy task. So people ask for 2 species, one rodent and one non-rodent, up and down methods, LD50 evaluation, which hardly we do. We just only claim that more than 2 gram we have given, more than 2 gram it is LD50. So we don't take per sex 15 to 30 animals. In none of the Ayurvedic study it has been taken. Per group 20. So we don't take it. So repeated dose, we have not worked for 9 months. For 1 year, we have not worked for. Minimum 2 satellite groups, we don't take it. Okay, the fact is that 10 times recovery group, we don't observe any significant pathology. That is why we don't go for second satellite group, recovery group. But still, people ask for, they expect for interim kills at 3, 6, 12 months. We don't do this. We take it as if we are giving, it is not recommended. Long-term administration is not, why not recommended, why we should go for 10-month study, 6-month study. But what is bad in this study, there will be a recovery group of satellite group of 6 months, after interim kill maybe at 6 months, at 8, 9 months. So you can have, and you can extend your safety till 9 months, 10 months, 12 months. It is not bad. It is not bad. It is not blaming our. It is not against violation of protocol that it is not recommended. Long-term administration is not recommended. So it is useless data. Why you are doing that? Because people are using. it is needed by the hook or crook. Either there are physicians are prescribing or people are consuming. By whatever the mean, it is there in the public domain, even from what platforms, I don't know, Amazon and all, people are procuring and consuming. So it is need of time to go for this higher standards. So 23 to 48 histopathology pairs, we are working with, we are not working with pairs. So both the kidneys, we are not working with that. So we should work with pairs, and a lot of data, evaluation of neurological, cardiac and ophthalmic needed, because you don't know the presentation. You should we should go for this documentation also. There are many lacunas in terms of safety evaluation. For clinical evaluation, we lack long-term prospective and retrospective safety study documentation. Then systematic monitoring, scientific documentation without missing data, inclusion of vital data, we don't include it. We don't include timelines, and systematic reviews, meta-analysis, documentations practices are not widespread. There is a lack of synchronization in conduction of preclinical studies. जो हमने सेफ्टी स्टडी जिसकी देखी है, उसी से हम आगे नहीं चलते हैं कि उसी की फार्माकोलॉजी देखी, उसी का सब कुछ दिया, उसी पर सब कुछ किया. We don't adhere to this protocol because there is lack of documentation of systematic documentation of the toxicity study in pharmaceutical development, pharmaceutical analytical development. So people opt for this. फार्मास्यूटिकल एनालिटिकल डेवलपमेंट किसी और का होता है, किसी और प्रोटोकॉल का होता है. सो देयर आर मेनी लैकुनास for documentation of safety, because if somebody is working with nephrotoxicity, neurotoxicity, somebody is working with genotoxicity, the same formulations we develop with same protocol. If one institute is working, then it is most likely that it will work on same protocol of same pharmaceutical analytical protocol. But if others are working, there should be synchronization, harmonization. Until and unless there is harmonization, it will be difficult to conclude safety documentation. And if there is discrete data, then putting it into one thread, it will be very difficult. For preclinical studies also, we do have several limitations that, as that I mentioned. We have age limit. We cannot work with more than 1 year. We cannot work for 2 years. If you are working, administering 1 year, then for 1 year more recovery, we don't have animals, and there are a lot of concerns in that. So you cannot work up with multiple formulations and things. So I know I have taken more, I think 10 minutes more, and I will wind up within not more than 5 minutes. So there are absolute challenges in improvement of documentation of safety of Rasaushadhi. As I mentioned, in vitro, it is outdated, because in results of in vitro, you cannot, because the for pre-metabolic and pre-metabolic interactions and after bioavailability, what happens, it is not replicated, solubilities. There are a lot of concerns. So that chapter is closed. I don't say that it is useless data, but least extra palatability. But still, rest of we have many things we have discussed here over. For clinical also, we have discussed a lot of things. There are challenges for long-term studies, because you cannot administer. People will ask how you are administering 1 year. So, and if you are going for retrospective data, it is poor quality data. There is lot of chances of lot of missing data, and there may not be digitalization. Likewise, there are many challenges. And in prospective data, no ethical, there will be ethical concern. If you are planning for 6-month administration, 1 year administration, 2 year administration, there will be ethical concerns and continuity, lifestyle change. Lot of things are there, and a lot of challenges are there, but still it is not impossible. So that what is possible, collecting data, collecting what ongoing practices, if you digitalize, if there are well established protocols for evaluation, and we have proposed the same, although it is underway to finalization, a lot of work has been done. So there are many challenges for other issue also, biological tissues, you cannot have data of distribution and all. So the way forward is upgradation of toxicity protocols, then evaluation of long-term toxicity study. You can evaluate longevity, in-depth mode of dying. Likewise, then evaluation of hepatoprotective. These are you can go with alternative ways. You can test hepatoprotective activity, nephroprotective activity, neuroprotective activity, cardioprotective activities, so as to document safety protocols. But there are again challenges, because this doesn't happen as it happens clinically. These protocols are with a single shot you damage there is cardiotoxicity, and the protocols which are there, those are not suitable to us. It is not a platform to discuss these things, but the protocols, it is very difficult to find a good protocol which will have clinical applicability. But still, we can go for these evaluations, which will generate data of their safety. We can collect the data of clinical studies for done on Shoth, on Kamala, where Rasaushadhis are done. It is also a good proof of clinical documentation. So we can work on other animals, small animals also, zebrafish likewise, and evaluation of parameters of other hepatic neurological in clinical studies. There can be data of follow-up, long-term clinical studies can also be there. And this is therapeutic drug monitoring. Whatever you are doing, prescription studies, consumption studies, whatever you are doing, practicing in vulnerable group, let it be in pediatric, whatever doses, which medication, whatever it may be, the company, mode of administration, whatever you are doing, if it is documented, it is also a good documentation. It gives a lot of signals for safety, but it is not there in situ. Right now we are not optimizing HIMS facility for digitalization and its documentation potential of therapeutic documentation. So there is we do have a lot of protocols, lot of studies, and lot of available protocols for evaluation of safety, but why still clinical documentation is needed? Because long-term follow-up and long-term administration is not possible, and there is question, always question on duration of administration of Rasaushadhi. So there is always variable use in clinical setup, and with different anupans, drug combinations in different modes, different pharmacies will prepare in different manner, and that is why you cannot go opt for these protocols, and you cannot have even prospective studies of clinical studies, well-planned clinical studies. Only the answer is if you capture the data of clinical safety from ongoing setup in a clinic available setup, ongoing practices, if you capture, that is a very good documentation of generation of clinical safety of Rasaushadhi. So it can be, as mentioned, in healthy volunteers. For Rasayan, people may opt for Rasaushadhis. For healthy individuals, for hair falls, for trivial errors, which are, although in most of the respects those are healthy, but trivial problems may be there, and they may go for Rasaushadhi administration. The same endocrinal variation in healthy, in this particular disease conditions where it alters the most and where it preserves. So this data can be there. There can be vulnerable population in D6PD deficiency, in history of where drug sensitivity is there, sulfur drugs. You can administer, and I hardly found only 1 or 2 cases here for allergy to Gandak Rasayan and Gandak Muller application. So there are G6PD deficiency patients, hypersensitivity to drugs, liver impairments, fatty liver, cirrhosis, chronic alcoholic. In those patients, there are clinical studies. We are prescribing daily to these patients. There are renal failure patients who are consuming. Documentation of long-term administration of Rasaushadhis can be there in many cases. There can be, as OTC medicine, there are several indications where people may either consume or people may administer, say in case of carcinoma, likewise, and for Amlapitta, Shwas, likewise for painkiller, as a painkiller, people may consume or people may prescribe. these medications, and that can be a good case for documentation of practice of 1 year, 2 year, 5-year administration. Let it be only few handful of 10 cases, but their documentation can be good. Diabetic nephropathy, diabetic neuropathy, worsening of diabetic neuropathy. It is well known fact that we administer a lot of mercurials, lead containing and arsenicals in post cerebrovascular episode. पक्षाघात में बहुत सारे फॉर्मुलेशंस है. Now it is well known fact कि जो पहले रिकवरी हुई है पेनुम्ब्रा हम सिर्फ प्रोटेक्ट कर सकते हैं. जो डेथ हुई है एक बार इसकी न्यूरल टिश्यू की, इट इज नॉट गोइंग टू गेट रिकवर एट ऑल. और जो कोई पेनुम्ब्रा को हम सिर्फ रिकवर कर सकते हो 10-15 दिन में. जो रिकवरी हुई बाद में तो जो मसल स्पास्टिसिटी वगैरह उसका जो रिकवरी हुआ वही वो रिकवरी हुआ 1 month 2 month में. आफ्टर 1 month 2 month it is foolish thing to expect recovery, but still people are coming, we are documenting, and we are doing lot of things. People are coming, we are documenting. Even after 6 months we are documenting improvement in neuropathy. So with your lead, with your mercury, there should be worsening, and if it is worsening, the patient should worsen. If you are administering this medication, patient should worsen. If there is worsening of 1%, what a contemporary neurologist says, there should not be a worsening of 1%, even 1%, because 1% worsening means there must be again next cerebrovascular evidence. Otherwise, there should not be worsening. If there is no worsening and there is improvement, it is a very good clinical documentation that in Pakshaghat, if you are administering this lead containing formulation, arsenicals, and only the documentation that it has not worsened is a very, forget about improvement. It has not worsened by 5% or 10% or even 1%. It is a very good documentation. But we are missing these documentations. In a diabetic nephropathy, there can be a documentation, and there is no documentation, although that was evaluated in Nagbasma, administration of Nagbasma in diabetic nephropathy. Nephropathy was recovered, but neither there is a publication in a public domain, neither there is a thesis. But I am evidence that these documentations were there. The 24-hour microalbuminuria was there with urinary creatinine measurement, and there were improvements in GFR. So in HG toxicity, as I mentioned, catecholamides should be there, MDA should be there, ACTH should be there, steroid should be there, all that can be. And because these are list of parameters, people expect that this gets altered, and this will get altered. People will take this as toxicity, but if it doesn't worsens your disease, if it doesn't worsens your health parameters, it is well and good. There is no, as you expect, the documentation of your neuropathy, documentation of your memorial intellect, neuropathy with nerve conduction studies, with renal parameters, more stringent renal parameters, hepatic parameters could be there. If you document that others are in well situ, let it be alterations may be there. In healthy, there may be alterations, but in disease, it is severely, wherever you are administering, say, catecholamides in hypertensive, there is significant drop in catecholamides. In healthy, there is alteration, but it is not altering a healthy state, and it is not in long term, it is not worsening. So this documentation may be there. So along with Patya, there could be documentation of safety. There are several safety documentation protocols which are just proposed, but there could be such a lot of what can be administered with which method, lot of list of parameters. There is a good list of parameters also. So that would, we will share and discuss in detail. Those are underway for their development, but there could be a lot of parameters, and it is needed. People ask for data of not only the hemosuppression or HB prevention or maintenance of HB. People ask for data of ALA, earliest manifestation of toxicity. Protoporphyrin 9, if you give the data of 3, delta ALA and protoporphyrin, they will accept this. If you give data for nerve conduction study that it has not been worsened, sensory, motor, it has not been worsened, it is good data. If you give the data of 24 hours protein urea, urinary creatinine, GFR, if you give the frequent data of GFR, people will then accept this one. So why to design protocols? So take home message is to there is need to upgrade evidence of safety of Rasaushadhi, safety evaluation through in vivo protocols. I know CCRS had given CDRI Lucknow to their planning, and it is underway with optimizing evaluation of safety. It is also underway. AIA Goa has recently conducted a series of workshops, and it is also paving the pathway for generation of data of review of safety. So I know a lot of people are doing, many people are doing, but it is not in one platform. It is not documented in well formed, and we are missing a lot of things which we can capture. So specifically for clinical documentation of safety. So with this, I have taken more than 20 minutes. आशा है कि सब लोग माफ करेंगे. And I am again acknowledge failure to prepare this PPT were not for this you audience, August audience, and I failed to put it concise, and there is no conclusive message. And initially I failed to acknowledge that I will not give any conclusive remarks, and there is nothing new. I already mentioned that these are my personal views, and it is a matter of debate, it is a matter of policy, it is a matter of regulation, it is a matter of lot of things, and lot of the people are doing many things. I am very small fry. I am not an authority in this field. Whatever my opinions were there, I shared it. I utilized this platform for giving me a good platform to communicate. I am very much thankful to all authorities of Vishwa Ayurveda Parishad, Namrata Joshi Madam, Agrawal Sir, Ramesh Gupta Sir, Adi Ji, and Shukla Sir, Mahesh Vyas Sir, and of course Director ITRA, Mesri Madam, for giving me this flexibility, free hand. My head, Professor BJ Padgiri, and utilizing data of institute. I accept for their acceptance for disclosure of many data. So on their behalf. So to my teachers and mentors, I pay gratitude, and not but least of the audience. Thank you. Thanks a lot.
Thank you, sir. Thank you.
Thank you so much, sir, for emphasizing on the importance of documentation in advance in Ayurveda. Because in this today's evidence-driven world, we truly if we truly want Ayurveda to flourish, we must prioritize the collection and analysis of our data. I request all participants to kindly drop your questions in the chat box, and I'll take them up one by one on your behalf.
I may not be satisfactorily answer a single question out of thousands, and as I am not an authority, but still I will put forth my little views, what I think. So please, you are free to.
Yes, sir.
There are
Sir,
lot of opinions, and I will just put forth my one among mine.
Yes, sir. I'm sure the participants found it enriching and thought-provoking, because I did. So moving forwards, I now request Dr. Ramesh Gupta sir from Pilibhit. Yes, sir. Dr. Sanjay sir. Hello.
हेलो. हाँ डॉक्टर
हेलो संजय सर. हाँ संजय सर.
हाँ बोलिए रमेश जी.
Okay. Thank you,
I
Dr.
now
Shiva.
request Dr. Ramesh Gupta sir from Pilibhit to deliver the vote of thanks and do
Thank you,
Shanti
Dr.
Path.
Shiva.
Okay, sir.
Good evening to everyone. First of all, I would like to thank our guest speaker, eminent trust scholar, Professor Prashant Babanrao Bedarkar sir, Professor, Department of Rasashastra and Bhaishajya Kalpana, Jamnagar, for enlightening us about their different safety parameters and method of safety evaluation of metals mineral preparations. The lecture was very much informative and purely based on your experiences in the field of toxicological research and your concern about the safe use of Rasaushadhi. I am pretty sure the precious knowledge delivered by you will definitely help in the building of confidence among us to conduct pharmaceutical and toxicity studies of different dosage pharmacologic medicines. I would like to express my gratitude to all entire respected teaching faculty from all over the country who have blessed and encouraged us with their presence. I am very much thankful to Dr. Bhaishajya ma'am from USA for his persistent presence in the every webinar of Rasashastra. My sincere thanks to all our participants, PhD scholars, PG scholars, and undergraduate students. This vote of thanks would be largely incomplete without expressing my gratitude to convener of this webinar, Professor Namrata Joshi ma'am from BHU, co-convener Professor Sanjay Agrawal sir from NIA, co-coordinator Dr. Aditi Kulkarni ma'am from Mumbai, and all our regional coordinators, Professor Upendra Jala sir from Nadiad, Professor K.L. Virupak Gupta sir from Chennai, Professor Akhilesh Srivastava sir from Indore, Dr. Piyush Chaudhary sir from Sonipat, and Dr. Suchi Mitra ma'am from Haridwar are working hard to make each and every webinar successful. I am very much thankful to Dr. Shivakshi Sharma for nicely moderating the session. At last, I wish my sincere thanks to all office bearers of Vishwavit Parshad who have directly or indirectly involved to make this webinar is a wonderful learning experience. Thank you all again. Namaskar.
Namaste.
Now we will recite the Santi part. For this, we sit straight and recite the mantra. Sarve bhavantu sukhina, sarve santu niramaya, sarve bhadrani paschantu, ma kaschid dukh bhag bhavet. Om Shanti, Shanti, Shanti. Thank you, Prashant sir.
Thank you, sir.
For
Thanks
your
for giving
very much energetic lecture.
Yeah.
Thank you, Dr. Prashant ji.
Dhanyawad, sir. Dhanyawad.
You presented your view regarding the safety of Rasaushadhi. This is a big challenge, I know. Thank you. It's a very informative lecture.
Thank you, sir. It was a really nice lecture.
Thank you, sir. Thank you.
Thanks to Dr. Shivakshi Sharma also for moderating this session.
Thank you, sir. Thank you, sir.
Okay, sir.
Okay, sir. Okay. Thank you.
Okay, good night, sir. Good night. Good night to everyone. Thank you, sir.